The laboratory today is concerned with hematopoietic malignancies, that is, lymphomas and leukemias. And to start out, you will be examining several types of lymphomas. I would like to remind you that lymphomas arise from neoplastic lymphocytes and occur in the lymphoid organs, including the lymph nodes. However, before you can recognize what the pathological condition looks like, you must first familiarize yourself with the normal structure of the lymph node. For this, place the slide labeled :6 on a piece of white paper and examine it by eye. Select one of the normal nodes that does not contain a metastasis, as indicated here, and center this on the microscope stage. Examine the normal node under the 4x objective lens. Identify the capsule, which is composed of a thin layer of collagenous connective tissue, the subcapsular sinus, which is located immediately below the capsule through which the lymph flows, the lymphatic nodules, which are composed of masses of lymphocytes, and the germinal centers, which are the lighter staining areas inside some of the nodules. And finally, note the size of the germinal centers relative to the entire node. This will be important later on in distinguishing normal morphology from pathology. Now switch to higher power and observe one of the nodules with a germinal center. At the periphery, you will see the lymphocytes, which appear as small basophilic dots that are composed of condensed nuclei with just a thin rim of cytoplasm. Within the germinal center, the majority of the cells are immunoblasts. These are lymphocytes that have been activated. They have migrated to this region, expanded both their nuclei and their cytoplasm in preparation for cell division. In addition, there are other cells, such as macrophages, which are large cells with a light-staining cytoplasm, and plasma cells, which I will describe later. As indicated in this chart, lymphomas can be classified into two major types: Hodgkin's disease, of which we have two examples, and non-Hodgkin's lymphomas, or NHLS for short, of which we have one example. Now, the defining feature of Hodgkin's disease is the presence of Reed-Sternberg cells, or RS cells for short. These are the malignant cells that are believed to be derived from the B lineage of lymphocytes that also give rise to antibody-secreting cells. As illustrated in this figure, the RS cells come in a variety of shapes and forms. Now, the classical morphology is shown in the upper left-hand corner here. They are generally large and often have two large nuclei that contain prominent nucleoli. Importantly, they are often represent only a small fraction of the cell population. Most of the other cells that surround them are normal cells that have been recruited by factors secreted by the RS cells. Now, in this laboratory today, I am requesting that you at least attempt to locate an RS cell. Examine the slide labeled Hem1 by eye and observe that it contains two samples of lymph nodes. Note that one of the sections contains a rather thick eosinophilic capsule and thick bands of collagen within the body of the gland. This is indicative of the nodular sclerosis subtype of Hodgkin's disease. Normal nodules with germinal centers can be observed in the smaller section on the left, but are reduced or absent in the larger one on the right. Please note the location labeled area of defacement, where there are very few nodules. Now place the slide on the microscope stage and examine the smaller section under lower power. As illustrated here, you can see numerous nodules with germinal centers. Now move the slide to the larger section and center it on the region labeled area of defacement. Observe that nodules are rare or absent in this location. This is a region that the RS cells are easiest to locate. Also note the thick bands of eosinophilic staining collagen fibers. Switch to medium power and observe the large bands of collagen that are indicative of the nodular sclerosis subtype. Scan the region of defacement for large, dark staining cells that have a clear area around them due to shrinkage. These may be the Reed-Sternberg cells that are diagnostic of Hodgkin's disease. When you locate a likely candidate under low power, center it and switch to higher power. This is an example of a classical RS cell. It is large, has a dark staining cytoplasm. It has 2 nuclei and prominent nucleoli. However, these cells are rare and hard to find. I recommend that you spend no more than about 5 minutes looking for this cell, since not all the sections will contain a classical RS cell. Now, as I said before, most of the cells in the section are normal and consist primarily of lymphocytes with a smaller proportion of macrophages. There are also eosinophils. These are a type of leukocyte that contain small eosinophilic granules and lobulated nuclei. So they can resemble RS cells but are much smaller. Also note the plasma cells that secrete antibodies. These cells have a dark staining cytoplasm, and the nucleus is off to one side, and it has the appearance of a cartwheel. The slide labeled Heme 2 is another example of Hodgkin's disease. However, it is of a different subtype. It is of the mixed cellularity subtype and does not have the prominent bands of collagen found in the nodular sclerosis subtype. By eye, observe that this slide contains a single node with a thin capsule. Under 4x magnification, note that no nodes or germinal centers are present, indicating diffuse defacement of the normal architecture. However, there are broad regions containing different cell populations. Switch to intermediate power and observe the different cell types that are present. In this figure, you can see a dark staining region containing a concentration of lymphocytes. There are other regions of lighter staining histiocytes, which is simply another name for macrophages. This is indicative of the mixed cellularity subtype. Now, scan the area at low power, and when you locate a structure of interest that could be an RS cell, switch to higher power. This shows a classical Reed-Sternberg cell. This one is large, binuclear, and has distinct nucleoli. Since these classical RS cells are hard to find, do not spend too much time looking for them. Now, the second type of lymphoma are the non-Hodgkin's lymphomas, or NHLs for short. These lack the RS cells. Examine the slide labeled Heme 3 by eye and observe that nodules are present throughout, although they are hard to see without magnification. This is an example of non-Hodgkin's lymphoma of the follicular subtype. Under the microscope, observe that the follicles are present, but they appear abnormal. The germinal centers are much larger than normal. This slide is a side-by-side comparison of the nodules from a normal node and that from a lymphoma. The colon 6 slide is shown on the left, and that from the follicular lymphoma is shown on the right here. Clearly, the germinal centers of the lymphoma occupy a greater proportion of the nodules than that from the normal node. Now, large germinal centers can occur normally when a node is undergoing a large immune response, or abnormally in the case of a lymphoma. Now switch to higher power and examine the germinal center. This slide shows a high-power view of the germinal centers from a normal node on the left and that from the follicular lymphoma on the right. Please note that while there is much diversity between the cells in the normal node, the cells from the lymphoma are much more uniform in both size and shape. Indeed, they represent a monoclonal expansion of the malignant cell type. Now, please continue on to the video labeled TBIO 572 Hematopoietic Malignancies, Part 2B.